Hi everyone, and welcome to our video on the function of the kidney. This is actually going to be split up into two parts on the different aspects of functioning of the kidney, because otherwise it'd be quite a lengthy video, to be honest. What we're going to be doing in this video is of course having a look at the specification reference 5.1.2c part 1, which is the structure, mechanisms of action and functions of the mammalian kidney. What we're going to be doing in this video then is having a look at two of the parts of the kidney function itself. We'll have a look at ultrafiltration and the selective reabsorption. Part 2 of the function of the kidney will then look at the reabsorption of water. So, ultrafiltration first of all. What we saw in the previous video on the structure of the kidney was that we've got these two arterioles. We have the afferent arteriole, where the blood is going to be arriving to the glomerulus, and the efferent arteriole, where it then leaves. Now, what we do have is a difference in diameter between the afferent and efferent arteriole. So what we have, remember, is a wider diameter on the afferent and a narrower diameter on the efferent. So because of that, what we're going to see is a build-up of pressure. So we're increasing the pressure in the glomerulus, and therefore we've set up a pressure gradient. And this then means that we're going to be pushing fluid out from the capillaries, obviously through those three layers we looked at last time, into the Bowman's capsule itself. One thing that we need to understand in terms of how this pressure is all set up and how these fluids are going to be moving is a little bit of a recap on two types of pressure we encountered back in module 3, where we were looking at the formation of tissue fluid. We've got hydrostatic pressure and oncotic pressure. Hydrostatic pressure is going to be caused by the blood, it's basically blood pressure in this case. And then we've got the oncotic pressure, which is down to the plasma proteins. So hydrostatic pressure of the blood is going to be acting to force fluid out of the capillaries. But because obviously we've got these proteins in the blood, which are going to be too large, greater than 69,000 molecular weight, that therefore can't leave our actual capillaries, then we actually set up an oncotic pressure as well. So we've got these two opposing pressures, just like we did in the tissue fluid. The filtrate itself in our Bowman's capsule has hydrostatic pressure and oncotic pressure of its own, but both are low in that case. So what we're really doing here is when we look at all four of those pressures, we've obviously got quite a high hydrostatic pressure coming from the capillaries towards the Bowman's capsule. We've got a smaller oncotic pressure going in the opposite direction. And then in terms of the filtrate, we've got a small hydrostatic pressure and a small oncotic pressure. Because both of these are low, then when we have a little look at the overall pressure, it's acting in the direction to force substances from the blood into the filtrate. And that's predominantly driven by that hydrostatic pressure caused by the structure of the kidney, with that narrower efferent arteriole and the wider afferent arteriole. So as a result of this net pressure that's then moving from the glomerulus into the Bowman's capsule, we are going to be forcing blood plasma out. And that blood plasma obviously contains an array of different substances. We've got water, we'll have things like glucose, urea, inorganic ions, amino acids, all of those substances that are small enough, i.e. less than a molecular weight of 69,000, they're going to be moving into the Bowman's capsule. Any of those blood cells and large plasma proteins are going to be left in the capillary. So anything with that molecular weight greater than 69,000 remains in the capillary itself. And as a result of that, we find that the blood has this very low or very negative water potential. So that means we're not going to lose all the fluid from the blood, because obviously we've got that oncotic pressure, which is going to be drawing a small quantity of fluid in at that point. So we won't lose it all. There's always going to be a certain proportion of fluid left in the blood, otherwise we'd just be left with a lump of cells and large proteins, which obviously wouldn't be good because that's basically a clot. So that was ultrafiltration then, where we're going to see certain substances obviously being able to leave the glomerulus and enter the Bowman's capsule if their molecular weight is small enough. Anything with a large molecular weight is going to remain in the blood. So it's a filtration process because only small substances are actually passing through into the filtrate. The second stage of our kidney function is the selective reabsorption phase. So once we've got that filtrate in the Bowman's capsule, it then passes into the proximal convoluted tubule, or the PCT. Now in the PCT, all of those sugars, so all the glucose, most mineral ions, and some of the water is going to be reabsorbed. So what we're seeing here is we don't want all of these things that we have pushed out of the actual glomerulus into the Bowman's capsule to just end up lost in the urine, because that would be incredibly wasteful, and therefore that PCT is going to reabsorb certain items, all of the sugars, most minerals, and some water. And the way that these cells are going to be specialised for this is that, as usual, they're going to have this highly folded surface, making these microvilli, so these small little protrusions, and therefore we've got a very large surface area. So thinking about those standard things that we talk about as far as exchange surfaces go, large surface area, typically caused by increased folding, is one of those key principles that we've looked at time and again. So what we actually refer to is a thing called a brush border, which is basically just that surface of those cells which have got all these little microvilli, and when you look at them under a microscope it does kind of look like the surface of a brush, to be honest. When it comes to the actual process of reabsorption, then we've got two key aspects to keep in mind. We have active transport, and if we drop back to our module 2 knowledge, clearly this is a process that is active, so it involves ATP. And then we've got co-transport as well. Now, if we think of the word co-transport, then if you think of something else, cooperate, for example, that's where things work together. So co-transport is where we're going to be moving things together. So what we actually have on that cell surface membrane here are co-transporter proteins, and what they are going to be transporting are glucose or amino acids, and they're going to be moving them alongside sodium ions. So if we have a little look at the diagram on this side, we can see in the middle there, this little section, we've got our little co-transporter. That's the little blue circle we can see there. Now what we can see is amino acids or glucose are on our little red line, and then we've got our sodium ions on the little green arrow underneath. So what's actually happening is we are moving the amino acids or glucose alongside the sodium ions through that co-transporter protein, and they're going from the tubule into the cell itself. Now, what we're going to find is that in order for this to actually be carried out, we need ATP. And the ATP is going to be used in another pump, the sodium-potassium pumps. So what we're going to see here is we then need to utilise that ATP to actually cause those sodium-potassium pumps to work. So what happens is we've got our little sodium ions that are obviously going to be accumulating inside the cell, and we then need to basically pump them out because we don't want them accumulating inside the cell. So what we do is we use the sodium potassium ion pump to actually move those sodium ions from inside the cell into the capillary itself. As a result of that active process of pumping the sodium ions into the capillary, then we decrease the concentration of sodium ions in the cytoplasm of the cell, so we've set up a concentration gradient. That means the sodium ions are going to be diffusing down their concentration gradient into the cell through that co-transporter protein, and as they do so, they take glucose or amino acids with them. So what we actually have here are these two different proteins set up in the membranes in order to move our substances across. The way in which this co-transporter protein is going to work then is that it actually has binding sites for those two substances, the sodium ions and either glucose or an amino acid. So when we actually have both of those binding sites occupied, then the protein is going to change shape. And as a result of that change of shape, it's going to then deliver those sodium ions and the amino acid or the glucose to the cytoplasm of the cell. So it's purely relying on that change of shape of the protein to then move those substances inside. If we just take a moment then to consider these adaptations that we've mentioned throughout this, and obviously some that hopefully we've made some assumptions on from our prior knowledge, then those PCT cells are going to have these microvilli for the large surface area. And obviously any of these surfaces where we're having these exchanges need a large surface area, because the larger the surface area, the more space we've got to pack in all these different transporter proteins. Second thing they're going to require are lots of mitochondria. And hopefully this was an assumption we made because we've been talking about these active processes. Any of these active processes require ATP. In order to produce ATP, we are going to require the mitochondria to carry out respiration. We then also have rough endoplasmic reticulum in there, because the rough endoplasmic reticulum is the site of protein production. So that's going to be where the sodium potassium ion pumps are made, the co-transporter proteins, other carrier proteins. All of those are going to be manufactured within the rough endoplasmic reticulum and then obviously inserted into the cell surface membrane there. As always, I hope you found this video useful. Don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website where you can find an array of other resources to help you in your A-level biology studies.